Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing life science question paper conducted in the year 27th June 2023. This is part 1 of the explanation. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers being answered for the life science subject, you can go to my channel Omni Bioentrance. Go to the playlist, search for CUET and you will find life science question papers of previous year, still previous year question papers being answered there. Coming to the first question. Molecular absorption spectroscopy based on electromagnetic radiation in the wavelength region of. So it is 190 to 800. This is the same in option 2 also. So both the options are correct. 190 to 800 nanometers. The dye used for visualization of DNA or RNA in agarose gel is ethidium bromide. Answer is option 3. The annealing temperature during the PCR reaction should be. So annealing temperature is the temperature at which the primers bind to their complementary target DNA sequences. And this binding of the primers with the DNA sequence is via the hydrogen bonds, and this allows for the polymerase enzyme to start synthesizing new DNA strands from this primer. So the annealing temperature during PCR reaction, it should be 45 to 65 degree Celsius. So answer is option 2. Then size exclusion chromatography is used to separate the proteins on the basis of. So in the name itself it is given size exclusion chromatography. So it is based on the size that proteins are separated, and size exclusion chromatography is also called gel filtration or gel permeation chromatography. Then next one, in complement systems, which is not an example of anaphylotoxin. So anaphylotoxins, they are fragments which are generated from the complement system, especially from C3, C4 and C5. That is like C3A, C4A, C5A, or these are anaphylotoxins. And what do these anaphylotoxins do? They cause smooth muscle contraction, increase vascular permeability and release histamine from the mast cells. And this results in allergic reaction. But here out of these they have asked which is not an example of anaphylotoxin. So it is C7, that is not an example of anaphylotoxin because it is a part of the membrane attack complex, not an example of anaphylotoxin. So the answer here is option 3. Then next one. What are anti-isotype antibodies? So anti-isotype antibodies are recognize specific isotopes of the immunoglobulins. Then isotypes of immunoglobulin that are found in human beings are IgA, IgD, IgE, IgG, IgM. So these anti-isotype antibodies, they recognize and bind to specific isotypes of hemoglobins. Even there are subclasses for some hemoglobin molecules, like IgG has IG1, IG2 like that, and IgA has IgA1, IgA2, IgA2. So these antibodies, they can also bind to these different antibody subclasses. So they are called as anti-isotype antibodies. And anti-isotype antibodies, they are specific for heavy chain constant region and light chain constant region. Not variable region but constant region of both heavy chain and light chain. So as per the options given here, option 4, if you see, directed against determinants present on constant region of a particular heavy or light chain class of antibody, but not to any of the others. That is correct. Answer is option 4. If you see option 1, it is directed against antigenic determinants of particular antigen combining site of a particular antibody. That is what they mean is that it should bind to the antigen binding site, but it is not correct. Directed against antigenic determinants that are allele specific, that is also not correct. Directed against determinant present on the variable region of the light chain class of antibody, not variable region, constant region of both heavy and light chains. So option 4 is more correct. Then next one, which molecule does not take part in inflammatory response? So first option, that is dopamine. It doesn't take part in any inflammatory response. It is a neurotransmitter and it is involved in various physiological processes. So the answer here is option 1. Then next one, varicella zoster vaccine is administered for, so it is against chickenpox virus. Answer is option 3. Then next one, which microbe is not capable of degrading petroleum hydrocarbons? So it is Aspergillus that is not able to degrade petroleum hydrocarbon compounds, whereas all the others are given in the options can degrade. Aspergillus is usually, it is degrading cellulose, starch, protein, then also involved in antibiotic production. Then next one, prokaryotic mRNAs contain a conserved polypurine tract consensus AGG located about 7 nucleotide upstream from the AUG initiation codon. This conserved hexamer is called. So it is a prokaryotic mRNA and it is found upstream of the AUG initiation codon. So it is a Shine-Dalgarno sequence. Whereas Cossack sequence, it is found in eukaryotes. It is a eukaryotic translation initiation sequence and it is found surrounding the AUG start codon. Then consensus sequence means it is a general term for conserved DNA RNA sequences. Then Tata box, as you all know, it is a DNA sequence found in eukaryotic promoters. And it is needed for transcription initiation. The next one, which type of enzyme is known to remove methyl or ethyl groups from guanine bases that have been mutagenized by alkylating agents? So DNA alkylating agents, they are chemicals that add either methyl or ethyl, or I can say in general an alkyl group to DNA bases, and it results in mutations or cancer. So examples include nitrosamines, nitriureas, etc. And the enzyme that removes this methyl-ethyl group from guanine bases that have been mutagenized by alkylating agents are called as O6-alkyl guanine DNA alkyl transferase. I'll repeat, O6-alkyl guanine DNA alkyl transferase or AGT. Okay. So here they have not given the full name, they have given just alkyl transferase. So your answer here is option 3, that is alkyl transferase. And this AGT, it is also known as O6-methyl guanine DNA methyl transferase or MGMT. And what do these enzymes which remove this methyl-ethyl group do is, this is just for your information in case they ask next time. So these enzymes, they transfer the alkyl group from guanine base, that is the alkyl group from guanine base, they transfer it to their own active site cysteine residues. So this enzyme in their active site, they have got a cysteine residue. And these transfer this methyl or ethyl group from the guanine residue to the cysteine residue, then remove, that is how they remove the alkyl group, restoring the original guanine base. Then next one, name the technique Matthew Michelson and Franklin Stahl used to evidence DNA replication is semi-conservative. So this everybody knows. It is cesium chloride gradient density centrifugation. Density gradient centrifugation. Answer is option 1. Then next one. Two highly inbred strains of mice, one with black fur and the other with grey fur, were crossed. So inbred means they are homozygous. And all the F1 offsprings have black fur. So F1 offsprings having black fur means black is dominant. Here only one characteristics they have mentioned, that is color of the fur. So here F1, the black is dominant character. Predict the phenotypic outcome of the intercrossing of F1 offspring. So when you cross F1 with F1, what is the result that you get as the phenotype? So here I already told you they are inbred strains. So they are homozygous, black is dominant. So capital B for the black parent and small b for the grey parent. Upon crossing the parents, you get F1, that is black fur. Now you intercross the black fur and you get 3 black and 1 grey. These are the black individuals and this is grey individual. So they have asked for the phenotypic ratio. So it is 3 black fur and 1 grey fur. Answer is option 4. Then next one. How many number of autosomal linkage groups have human somatic cells? So in somatic cells of humans, how many autosomes are present? There are 22 pair of autosomes. The others are the sex chromosomes. So the answer here is option 2, that is 22 pairs. Then, in DNA microarray technique consists of large number of double-stranded DNA, small number of single-stranded DNA, large number of single-stranded DNA, large number of double-stranded DNA. So, DNA microarray, it involves immobilizing thousands of single-stranded DNA probes onto a solid surface. Okay, so thousands of single-stranded DNA probes onto a solid surface is being immobilized. Then you hybridize these probes with the DNA sample. Then you measure for the fluorescence or the chemiluminescence to detect for hybridization. So the answer here is option 2, that is large number of single-stranded DNA. Answer is option 2. Then next one. Small circular DNA such as plasmids and some viral DNAs, when have no breaks in either strand, are referred to as. So nucleosome. Nucleosome, it is a complex of DNA wrapped around an histone octamer. So it is not having anything to do with breakage in either strand. Then nicked DNA. Nicked DNA, it is DNA with single-stranded breaks or nicks. Closed circular DNA, yes, that is without having breaks in both the strands of the DNA. So that's what they have asked. So the answer here is option 2, closed circular DNA. Chromatid, everybody know, one of the two identical sister strands of DNA joined at the centromere part of a chromosome. That is called as chromatid. Then next one. Telomeres end with multiple repeated sequence of the pattern. So telomeres have T, T, A, G, 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 N repeated N times. N is the number of repeats. So here if you see, only option 1 is having that sequence correct. So it is T, T, A, G, G. So first one, that is option 1 is correct. Then DNA binding proteins associated with bacterial chromosomes are. So they are HU and H1. HU is histone-like protein from strain U93 and it functions in DNA bending, wrapping, compaction, etc. And H1, it is also called as HNS or histone-like nucleotide structuring protein and it functions in DNA compaction, gene regulation, nucleotide organization, etc. So the DNA binding proteins associated with bacterial chromosomes are HU and H1. Then eukaryotes have two major chromatin proteins in SMC family. SMC is structural maintenance of chromosome family. They are, so it is cohesins and condensins. Whereas cohesins, they hold sister chromatids together during mitosis and meiosis and they will ensure proper segregation of the chromosomes. And condensins, they compact the chromosome during mitosis and meiosis. The name itself suggests that they condense. So compact chromosome during mitosis and meiosis and they maintain chromosome structure and organization. Integrins, they are cell adhesion receptors and not chromatin proteins. Then lamins, they are nuclear envelope proteins and not chromatin proteins. Hemoglobin and myoglobin, they are oxygen binding proteins in RBC and muscles. Then actin and myosin, they are cytoskeletal proteins involved in muscle contraction and cell movement. So answer here is option 2. Then next one. In general, a carbohydrate molecule with N chiral center can have how many stereoisomers? So it is 2 raised to N. Now if you take glucose C6H12O6, it has 5 chiral centers and it has 32 stereoisomers. 2 raised to N. 2 raised to 5, that is 32. So the answer here is option 4. Then next one. The most stable structure for a random sequence DNA molecule under physiological condition is. So they have asked specifically under physiological conditions, that is at neutral pH, ionic strength, temperature, at physiological temperature, all these parameters, which is the most stable structure of DNA molecule. It is B form of DNA. So the answer here is option 4. Now A form, it occurs under dehydrating conditions and Z, it occurs under high salt, acid or alternating purine pyrimidine sequences. So it is not under physiological condition, both A and Z form. So the answer here is option 4, that is B form of DNA. Then next one. Porins are proteins found in gram-negative bacteria such as E. coli and allow dash to cross the outer membrane. So porins help in polar solute crossing. So the answer here is option 4. Then next one. In the enzyme ATP synthase, F0 domain is sensitive to. So ATP synthase, it is having F0 and F1 domain. So out of this F0 domain, it is sensitive to which antibiotic they have asked. They have given oligosaccharide and oligonucleotide also, but we will see for the antibiotic, that is oligomycin. Answer is option 3. And this binds to the F0 domain. This oligomycin, it will bind to the F0 domain of mitochondrial ATP synthase and it specifically binds to the C subunit of the F0 domain. Remember that C subunit of the F0 domain. And because of this oligomycin binding, what will happen is there will be block of proton flow through the F0 domain of ATP synthase, and this prevents ATP being synthesized and thereby inhibits oxidative phosphorylation. Then next one, which is not the type of positively charged amino acid? So they have asked which is not a positively charged amino acid. Arginine, histidine, lysine are positively charged. Whereas asparagine, it is polar and uncharged. So the answer here is option 3. Then next one. In most higher eukaryotes, the fatty acid synthesis occurs in cytosol. But in plants, it occurs in the chloroplast. Answer is option 4. Then next one. When the bacterium Agrobacterium tumefaciens comes in contact with the damaged plant cell, the segment of Ti plasmid called tDNA is transferred from plasmid and integrated at the random position in one of the plant cell chromosomes. The size of tDNA is, so the Agrobacterium tumefaciens, it is a natural genetic engineer. It will come when it comes in contact with the damaged plant cell, what it will do is, it is having a plasmid called as Ti plasmid inside it, and this Ti plasmid is having tDNA region between its left and right borders. It will transfer the tDNA into the plant cell chromosome, and this tDNA, it will get integrated at a random position in the plant cell chromosome. Now what the question is, what is the size of this tDNA in Agrobacterium tumefaciens Ti plasmid? So the answer here is, it is 23,000 base pairs. Answer is option 3. Then next one. The completed sequence of the human genome was published in, so it was published in 2003. Then next one. Plasmids are circular DNA. Of course. Naturally occurring bacterial plasmids range in size from, okay. So they range in size from 5,000 to 4 lakhs base pair. Answer is option 1. Then next one. Gene expression cannot be analyzed using the technique. Of course, DNA fingerprinting cannot be used for gene expression analysis. And gene expression analysis means studying gene transcription and translation. With DNA fingerprinting, we are not studying any DNA transcription and translation. Whereas it is a technique, DNA fingerprinting, it is a technique used to identify individuals or biological samples. And they will analyze certain DNA patterns, not transcription and translation. And it is used in forensic studies, paternity testing, and genetic in some genetic identity verification studies. So it is not used in gene expression analysis. Whereas fluorescence in situ hybridization or FISH, it visualizes specific DNA or RNA sequences within cells. Then RT-PCR, it measures mRNA levels to study gene expression. DNA microarray, we just discussed that. So that is also involved in gene expression analysis. Then next one. The most efficient and widely used method for transforming animal cells rely on, so it is liposome or viral vectors. Agrobacterium tumefaciens is plants transformation. Plasmids for mainly used for bacterial transformation. Even others mainly it is used for bacterial transformation. BAC is a bacterial artificial chromosome. Then next one. Which of the following phases in bryophytes is or are found freely in nature? So the gametophytic phase, it is found freely in nature. Answer is option 1. Which of the following plant has rhizophore? Now rhizophore, it is an organ that functions as a precursor to roots, and it provides anchorage, water and nutrient uptake. So it is a primitive characteristic and it is found in Selaginella. Answer is option 2. Then vascular bundles in monocot plants are, so arranged in rings. No. Arranged in stellar region. No. Scattered in stellar region. Yes, that's correct. Closed and scattered in stellar region. That is more correct. Closed means there is no cambium present between xylem and phloem. Then next one. Turpentine, which is an oleoresin, is obtained from. So it is obtained from pinus. Answer is option 1. Then protochordates are included under chordates by the presence of. So notochord. Answer is option 2. Scaly anteaters are included under eutheria. Answer is option 1. Hormones are released in. So hormones are released into the blood. Answer is option 2. Nerve impulse in a neuron travels in one direction, two direction, multi-direction from cyton to axon. That is more correct. So nerve impulse in a neuron travels in from the cell body to axon, cyton to axon. Then cataract of human eye defect refers to shrinkage of eyelids, wrinkling of eyelids, denaturation of protein in eyelids, denaturation of crystalline protein of eyelids. That is more correct. Answer is option 4. Water from digested food is absorbed from the large intestine. Answer is option 2.